Hello everyone, this is a continuation of the previous video on the role of red blood cell derived vesicles, and today we'll be looking specifically at their application as drug delivery vehicles. Now, as you can see from this diagram right here, RBCEVs are equipped with a unique lipid bilayer structure that enables them to encapsulate a diverse array of therapeutic agents. Their lumen can accommodate water-soluble drugs, while the hydrophobic regions within the lipid bilayer effectively sequesters the lipid-soluble drugs. Now, as you can see from this diagram of the lipid bilayer, the hydrophilic heads create what is an aqueous core where the water-soluble drugs can be stored. The hydrophobic tails, on the other hand, holds lipid-soluble drugs. Now, we'll be looking at a couple different methods of drug loading. First is passive encapsulation, wherein drugs diffuse into the vesicles based on the concentration gradients. Now, in this method, the hydrophobic drugs are fixed within the hydrophobic tail region of the lipid bilayer, whilst the hydrophilic drugs are pushed within the aqueous core of the exosomes. Second method is active loading techniques. Now, this is employing methods such as perhaps electroporation or co-incubation to force the incorporation of drugs, especially used for loading macromolecules like RNAs or DNAs into these RBCEVs. Now that we know the different mechanisms for drug loading, let's take a look at the targeted delivery of these drugs. Now, red blood cell derived extracellular vesicles, or RBCEVs, have a natural ability to target specific tissues, also known as tissue tropism, particularly notable within the liver and the brain. Now, this targeting ability is primarily due to the different specific interactions that occur between the proteins and the surfaces of the RBCEVs and the receptors that are found on these cells of the target tissues. First mechanism of targeted delivery is in receptor-mediated transcytosis. This facilitates the crossing of the blood-brain barrier, which allows the RBCEVs to deliver the neurotherapeutics directly to the brain tissues. Second method is in selective hepatic uptake. Now, this is for the liver specifically, and it's driven by surface markers that bind to the liver cell receptors, which optimize the delivery for liver-specific conditions. Now, let's look at a couple of methods to enhance the targeting capabilities of these RBCEVs. Now, the most famous method for enhancing targeting capabilities is in surface modification, wherein we're attaching ligands or perhaps antibodies to the surface of the RBCEVs, which can therefore enhance the specificity of the target cells, which thus reduces the off-target effects and improves the therapeutic outcomes. Now, in terms of kind of comparing the advantages of these RBCEVs over synthetic nanoparticles, these RBCEVs can distinguish themselves from the synthetic delivery systems like perhaps liposomes or even nanoparticles through several key advantages. The first one being biocompatibility. Now, as natural components of the blood, RBCEVs are inherently biocompatible, which reduces the risk of adverse immune reactions, but also reduced immunogenicity. They evoke minimal immune responses, which is crucial for repeated administration in chronic therapies. And also, besides that, enhanced circulation half-life. Now, RBCEVs naturally evade rapid clearance from the blood system, which allows for a prolonged drug action. Now, the remarkable capabilities of RBCEVs are currently being explored in various medical fields, including oncology, neurology, and hepatology, where the targeted delivery mechanisms are significantly impacting treatment paradigms. In the case of oncology, we're leveraging RBCEVs for targeted chemotherapy delivery, potentially reducing the systemic toxicity associated with conventional cancer treatments. Now, in the case of neurology, we're utilizing RBCEVs in order to bypass blood-brain barriers and deliver neuroprotective agents directly to affected areas in neurological disorders. In the case of hepatology, we're targeting liver diseases with enhanced specificity and efficacy, thereby improving outcomes in conditions like, for instance, liver fibrosis. Now, let's take a look at this diagram right here. This illustrates the steps from the collection of the red blood cells, their stimulation to release the RBCEVs, the isolation of the drug loading of these vesicles, and finally, their administration back into the patient for targeted drug delivery. So it pretty much sums up what we've just discussed. Alright, that is the end of this video. Thank you for watching, and I hope you gained a better understanding of the red blood cell derived extracellular vesicles and their potential uses and applications.